Today, I am extremely excited to introduce Katie Galloway. Katie is the WM Keck Career Development Professor in Biomedical Engineering and Chemical Engineering at MIT. Her work has been really cool. I've been a fan of Katie's work for a little while now. I feel like what they do in her lab is super creative, and I think having such a diverse interdisciplinary background is just so fantastic, and has already started to bring a lot to the transcription of chromatin field. I'm really excited about reprogramming, and then also of course supercoiling, which we're going to hear about today, which is such a tricky problem, and I'm really excited about it. So Katie is also like really accomplished. So she has both an NSF Career Award and a Mira, which is like seriously goals. I'm very excited about that. And she's also won several awards, the BMES Cellular and Molecular Bioengineering Rising Star Award, Princeton's CBE Saville Award, Lecture Award. And she has a really great background in chemical engineering, where she did her PhD at Caltech, and she did her bachelor's as well at Berkeley in chemical engineering. So I'm really excited to have some synthetic biology and engineering to also provide like new fun foundational understanding of biology itself too. So thank you so much, Katie, for agreeing to speak today, and I'll let you take the floor.
Thank you so much. I'm really delighted to join you all. I've been especially fond of seeing the work that comes out of this series, and our interest in gene regulation comes from the perspective of synthetic biology, where we're trying to control how cells turn on and off transgenes specifically. And I think there's some interesting things that we'll share today about how we think gene regulation is happening and why these systems are kind of an interesting space to then start asking questions about how genes are regulated outside of the normal context, where we can parse things a little bit more finely and build things up. So our interest specifically, besides just turning genes on and off, comes from this interest in being able to do cellular reprogramming. So I won't be telling you too much about this today, but our motivation is that we'd be able to actually turn on and off genes in the context of primary cells. And we know that that requires then the ability to build transgenes and gene circuits that are going to be able to reliably turn on and off programs we deliver. And what I'll tell you about today is about how we're seeing increasingly that there seems to be an aspect of biophysical regulation that's essential in the consideration of how we design these circuits, and that ultimately we think will be important then for controlling the process of reprogramming. So altogether, the thing that has been really interesting for us is thinking about as engineers that the chromatin and the nucleus is really this dynamical system. And what I mean by that is it's highly integrated. You have all of the chemical effects, structural proteins, and then also mechanical forces integrated together. And these systems are moving dynamically across, of course, really fast timescales, but then long timescales, and then facilitating or impeding cell fate transitions. And so at this intermediate level, these mechanical domains, which are mediated by supercoiling, are a little less studied for the reasons that they're difficult to study, and they're also super dynamic. Whereas, all of these structure of the histones, I think, are much more understood and additionally TAD. So these structures seem to be a little bit more stable. But at the level of mechanical domains, supercoiling seems to be an influence on the structure of DNA and potentially then on gene regulation itself. Especially within bacterial systems, it's known that transcriptional bursting is influenced by DNA supercoiling, and specifically some really elegant experiments from SNZ back in 2014, looking at that. And additionally, even gene circuits built on plasmids show these context effects that are attributed to supercoiling. But of course, bacteria are quite different than eukaryotes, and most notably, we have a nucleus to contend with. We have all of these other features and structural proteins. And so could supercoiling be relevant? And I hope by the end of the day, I'll convince you that it's important to think about supercoiling and how we design at least synthetic gene circuits, and potentially think about the native genome. Our goal overall in being able to turn on and off genes and deliver transgenes is actually to do reprogramming. I won't be telling you about that today, but it's ultimately that our desire is to be able to then take primary cells. So we work a lot with 293Ts, but our goal is to be able to make programs that work in primary cells, reprogram them and be able to do disease modeling, screening, and one day actually cell therapies to be able to program and really make the vision of synthetic biology to program cell fate come alive in terms of therapeutics. What that then looks like then is we spend a lot of our time in this direct conversion process, taking primary cells, fibroblasts, to motor neurons, and then understanding that process, and then ultimately using a number of different synthetic biology tools to figure out how to guide that process. That integrates into a slightly simpler vision where the challenge is, all right, we have these gene networks. How do we actually build controllers that interface with these networks so that we can predictably and reliably actuate changes in cell fate? And so what that looks like, again, for us is taking primary cells to induce neurons. So we really care that our programs can actually function in primary cells where it's relevant, which is a notable challenge, as you'll see in a few moments. And to be able to mediate this change, this cell fate change, not just to turn the genes on, but we know that the levels of the transgenes that we deliver, these transcription factors, their individual levels are really important for whether you can actually get reprogramming in the rates at which you do. And individual transcription factors show specific effects on the reprogramming process. And I won't tell you about this today, but there's a bioRxiv print that you can go and check out on that, which is motivating for us in terms of thinking, are our levels going to be set appropriately based on the transgenes that we are delivering and how we've structured the programs? And so more of what I'll be telling you today is then thinking about how do we actually design these controllers that are going to interface with the cell and its endogenous regulation. And specifically, I'll be telling you about supercoiling and specifically how gene syntax, which is the relative order and orientation of genes, actually affects how these two gene systems are able to turn on or not. And so and this is especially important, we've seen in primary cells. And so kind of a motivating example for why we care, we were trying to do something very simple. We were trying to take a TET-inducible promoter and just use that along with this trans activator, which people have been using this since the 90s. It's constitutively expressed. We have an MGL here so you can see it. And basically just turn on this gene. And so we're doing this with lentivirus because we're going to be delivering this to primary cells. In 293Ts, if you make this virus, you can see that, yes, you can turn this gene on. Here's your green lantern on this axis. You have your ruby here. And you can see that in the cells that have high green lantern, you have some ruby expression, but it's a lot of mosaicism. And so we're like, well, maybe the virus isn't that great. What happens if we put it into the mouse embryonic fibroblasts, right? The primary cells. There's almost absolutely no response. And so that told us, it's not good enough to make something that's decent in 293Ts and take it to our primary cells. We need something that really performs. And so our present challenge looks like this, and we kind of joke that it's this, our synthetic biology in 293Ts looks great until we take it to our primary cells. And this presents an opportunity too, I think, in terms of asking the question, clearly, in MEFs, we know that genes can turn on. So what are our synthetic systems not doing that normal endogenous regulation is able to do? And so this is kind of the synthetic biology side of our lab, thinking through, okay, how do we, if we really understood gene regulation, we would be able to build reliable systems that can actually turn on transgenes at the levels we want, and not just do that, on off, but actually dynamically regulate the way we see in native systems. And this is very much one of the, I think, inspiring quotes that you see in synthetic biology all the time is, what I cannot create, I didn't understand. And so if you understood life, you could build it. And that's, again, something we think about. How we do that oftentimes, though, is actually then reverse engineering and thinking about what is happening in these systems and are there motifs or structures that could facilitate. Here you can see this is a zebrafish developing, and you can see that there's a lot of variation here, but ultimately the somites get formed regularly. And this somite formation event is actually requires tight transcriptional coordination between two genes, HER1 and HER7. And so there are structures out there that we think are actually important then potentially for engineering transcriptional coordination. If you look across the genomes of different species, and then start to look for how genes are organized. One thing you can see is generally, if you look at a browser, right, they're not all organized, spaced out neatly as you might hope. there's different spacings, there's different also then pairings between genes. And so looking at these, what we call syntax, tandem, convergent, or divergent pairs across different species, you can see that in yeast or in humans, the patterns and enrichment for these different pairings are different. And most notably, what's interesting, at least in humans, you see this enrichment of this divergent pair at relatively short intergene spacing. And so, again, this is pointing to us to this idea that there's this non-random patterns of structures, and those non-random patterns might be pointing us towards a function. And so what could that function be? One, again, interesting example comes from this zebrafish developmental pattern. So in the somite formation, you have this HER1, HER7 system. These are repressors that dimerize, and they are spaced by 12 kilobases, which is relatively close within the genome. And what's interesting is it's known that if you make a cis mutation, so taking out the copy of HER1 or HER7 on the same allele, you get perfect pairing, and also then this beautiful formation of this tail. The somites form exactly as you need. However, if you take and make the transmutation so that you're not actually getting perfect copies, then from the same allele, you break the transcriptional coordination between the two genes, and you fail to get this tail forming properly, meaning that the transcriptional coordination of the tail formation are tied then to the two pieces of these two genes being on the same piece of DNA. And so that's very interesting then, just generally to think, okay, can genome organization then explain how we can get to this precision transcriptional coordination? And that's a big question to answer. So we wanted to ask something simpler, which is like, how about if we just put genes in different ways? Can we get different levels of coordination and through these pairings? And so this is, again, going back to that sort of build life to be able to ask, do these structures confer specific advantages in terms of prescribing how the genes will turn on and off? Okay. So Chris started thinking about this problem and asking the question, how could this co-localization, change? And specifically through the model of supercoiling. So during transcription, supercoiling is introduced as the RNA polymerase is coming and binding then to the double helical polymer, melting it so you get to a single-stranded piece of DNA. As the polymerase is then transcribing along, you get overwinding ahead of that polymerase, underwinding behind. So this is positive supercoiling. This is negative supercoiling. This is called the twin domain model. And normally, when we think about building models of gene regulation, we don't think about that what we have here is basically a symmetry breaking event where now this strand is no longer, symmetric. You have then a bias that's been introduced into how subsequent RNA polymerases are going to bind. And so normally when we model gene regulation, we would imagine that the subsequent binding of any other polymerase in this region would have, isn't dependent upon the supercoiling density, meaning that anything can insert. That's obviously wrong. And this is sort of the insight that Chris had. He said, I think it's going to be harder for RNA polymerase to bind over here in the overwound region compared to the underwound region. And so he said, let's model this properly. And then he went and looked up some actual modeling work from magnetic tweezers experiments from Michelle Wong's group and said, I think this is the energetic penalty we need to apply if we're going to ask for a polymerase to bind here. And so this is positive energy, which means there's a penalty. And the RNA polymerase binding should be limited there. Whereas here it's underwound. Actually, can you guys see my mouse? I did not even realize it. I've been waving at things. Hopefully you can.
I can see it.
Okay, thank God. All right.
Sorry.
So RNA polymerase binding will be favored over here. So now we have this difference in bias of where polymerase should bind in this model. And what emerges from that then is that you have one gene, this is not very interesting, but once you have two genes on the same piece of DNA, you then are going to have, again, an asymmetry where the downstream binding will be limited and upstream it will be favored. And so when this gene is on, this should be reduced. And we call this supercoiling mediated feedback. And to model this, we went in and did some stochastic simulations. And I'll tell you a little bit about those so that we could ask the question, well, okay, I have two genes on a piece of DNA. They're relatively closely spaced, which is what we normally do when we go to build gene circuits. What happens if I put them convergent, divergent, or in either of these tandems? And what I'm going to do is I'm going to have a reporter, which is some constitutively expressed, is some promoter that's constitutive. And so it has some constant base rate. And what I would like to do then is ask, when I turn on an adjacent gene, how does that change the activity of my reporter? Because my reporter should just be constant. And I'm going to ask, how does that induction then bias the activity of this reporter? Here on this axis, you can see how supercoiling is going to propagate across this. This is, again, the result of the simulations here. As we turn on the gene, you'll start to see mRNAs come up. And then when you hit this gray bar, we'll turn on the second gene. So at first, again, in the stochastic simulation, you have RNA polymerase is loading. This purple tail is just the growing mRNA. First gene's on, it's bursting away. You turn on the second, and what you start to see is, as this gene is on upstream, it's driving positive supercoiling into the promoter region of the other gene, which is going to restrict the probability that that gene is going to be able to burst. And so what happens then is that given either gene is on, you have this either or inhibition of the other gene. And you can see that here as you have this either or behavior. Conversely, when we have a divergent syntax, again, your first gene is basically bursting, you have this increase in negative supercoiling, which when we turn on the subsequent gene, both genes are going to start to burst with greater probability and frequency. So the probability that this gene is on when this one is on is very, very, very high. And that is then, again, because you have this reduced penalty for RNA polymerase binding. Okay. So what's interesting, though, is when most of the way we've built things in synthetic biology is often in this tandem syntax, and this one worried us greatly when we saw it, because here you go, you have your downstream gene, it's bursting away, and then you go to turn on maybe a TET-inducible element or something, and what starts to happen is that upstream element drives your downstream gene to silence. And if this upstream gene is then dependent on this downstream, that's a big, big problem. And if you're building a circuit where both of these genes need to stay on, this system is inherently very, very fragile. So that then summarizes into, okay, we saw the dynamics. At steady state, if you were to look at the joint distribution, so the expression of either your reporter or your inducible gene, you can see that for the divergent, you get really high expression of both. For the tandem, you see a bias for whichever gene is upstream. And then for the convergent at the mRNA level, which is what we're plotting here, you see this smear, which I'll tell you is going to average out when you get to the protein levels and we have reporters that are stable. So the question people always ask is, all right, so these genes are affecting each other, but what if you space them out? Does that matter? Supercoiling diffuses really fast. So actually spacing them out for this upstream tandem, there's really no change. You're going to drive that downstream gene, or the upstream gene will stay on. The downstream gene will have a small effect, but it's really very modest compared to the silencing you get from that upstream gene. And so spacing doesn't really seem to work. There's a lot more actually hidden in this modeling, but I want to tell you guys about the experimental stuff that we've done to kind of validate this model. But you can go and check out this paper at Cell Reports. There's a lot in there, including like how to build toggle switches. But again, this is all modeling, and we really want to know, does this actually work in cells? We were really encouraged when we saw that there was this beautiful paper from Teneki Lenstra's lab and led by Hedda Patel, showing that yeast actually have the expected profile of transcriptional coordination. They looked at the GAL1, GAL10 system, and they put these, they use MS2 and PP7 imaging in divergent or tandem to look then at the initiation between these two genes, whether they're tandem or divergent. We would predict that you'd have this increase in correlation in the divergent when they're in cis, but not in trans. And that's exactly what they saw. It's like, it's only correlated when you have this cis design and not in trans. And then in tandem, on the same piece of DNA, there's a small burst here, but basically nothing compared to what you see with this divergent syntax. But as we know, yeast and humans have very different enrichments of syntaxes, so maybe their gene regulation is quite different. And we really do care about being able to look at how gene regulation works in primary human and mammalian cells generally. So we set up an experiment, and by we, I mean Christopher set up this experiment where he integrated different two gene systems into 293Ts using piggyback as our starting, and asked, okay, how does this reporter with an EF1 alpha promoter change expression when he induces BFP, which is controlled by this TET-inducible element, which allows us to turn it on. So in the predictions, what we would have is that we'd expect the divergent should increase, the tandem should reduce, and so should the convergent. And so when he actually went in and made these lines, what you can see is exactly that. The divergent goes up as you increase the DOX concentration, convergent is very stable, and the tandem drops as we would expect. So then the question is, all right, fine, you did it once. What happens over time? Is this stable? Is this silencing? What's actually happening? So he went in and ran a very long experiment, or a relatively long experiment, pulsing DOX. And you can see that we can turn on the reporter gene or the inducible gene for all of these different syntaxes. They look approximately the same when you normalize them out. But when you go and look at what's happening in tandem, you see again this inverse response to the upstream gene being active, and it basically mirrors that. Convergent, again, super stable, probably because we're integrating out the effects of RNA stability and the protein. And then actually divergent, you see again, it's mapping along just beautifully with the inducible gene. And so in this system where you have basically the expectation that these are all biochemically identical, we're getting very specific differences in these systems. And so just to summarize, again, you have biochemically identical systems that only differ by their physical orientation, or as we call it, syntax, getting very different levels of expression. And these effects are repeatable, reversible, and specific then to how we've encoded them. So that's with our DOX-inducible systems. We were then really happy when Ross Jones, who's at UBC and works in Peter Zanstra's lab, reached out to us and said, I think I'm seeing some of the positional effects that you're talking about with your modeling. And what he was seeing is he was trying to co-express with constitutive promoters, neon green and then tag BFP, just 2 genes. And what he saw was that, okay, his tag BFP is really well expressed and M neon green not so much. Maybe he thought that's just the issue of, M neon green and PGK are really weak promoters, or it's the position. Asked the question about the position, he switched them and he moved his PGK upstream and his EF1 alpha downstream. And you would imagine that these should completely overlap if there's no physical coupling between these systems. Or if they're completely coupled, this should almost flip across the axis. And that's almost exactly what you see, where you have this upstream dominance where the tag BFP levels are now substantially reduced and the M neon green is substantially increased. So further, he wanted to ask, all right, so I looked at that with an EF1 alpha downstream, and then I want to change my upstream gene to ask, does it matter? So when he adds in PGK, CAG, EF1 alpha, he wants to know how much does tag BFP decrease. So as we saw, very few cells are going to be active with the PGK there. With CAG, it's actually not quite as much inhibition. And interestingly, when he has 2 EF1 alphas, there isn't this upstream dominance, suggesting that it's actually specific to the type of promoter. What's interesting is if you look at all through the synthetic biology literature, the only time this tandem system works and is used is when there's 2 EF1 alphas. And so we think there's something special about the EF1 alpha promoter, and it might be that there is oftentimes an intron within this promoter. So this was enough for Casey then to go back and look at her TET-inducible system in Lenti and say, all right, my first design was tandem. That was obviously potentially now a very weak design in the sense that this RTTA, which is needed for this Ruby to be active, is going to potentially get silenced as we turn on this TRE. And again, that's just to remind you, that's what we saw, this mosaicism in 293Ts and absolutely no response in MEFs. So she said, what if I flip this around and put my TET-inducible element going in the opposite direction? What would happen? And what she saw was quite remarkable. She was able to get this beautiful, clean response in the 293Ts with this lentivirus, and really to be able to restore the response in MEFs. It's not perfect, but we can work with this. This is a lot better than what we had before. And I think that while I see these flow plots and get excited, I think it's much more compelling to see actually the microscopy images where you can see that the tandem system has some fluorescence. It's not completely zero, but it's nothing compared to what we get in the divergent. And so we can really have the control of being able to induce genes, again, in our primary cells, which have been so challenging. So just to summarize what I'm telling you about supercoiling and what we know so far. So this divergent syntax is able to amplify the reporter when you have induction of that adjacent gene, whereas we see silencing with the tandem. This effect is reversible, and therefore we think it's not yet epigenetic, or at least not super stable. And this tandem syntax seems to show this upstream dominance where the upstream gene is expressed more highly than the downstream gene, as we would predict. And then finally, when we build these all-in-one systems with DOX induction, the divergent system better supports activation and it does very well in MEPS. So this is what we then think is some of the evidence that supports this model of supercoiling mediated feedback. What's exciting about it is it gives us a lot of room to be able to then control transgene levels depending on how you choose your syntax and how much you induce any one of your adjacent genes. The other thing that I think is really exciting about this is this system is entirely dependent upon the activity of transcription. So it is the process of transcription itself, not the products, that are actually changing what's happening to the adjacent gene, which is really exciting. And it's really exciting for me because one of my favorite topics, besides just thinking about reprogramming and other gene circuits, is thinking about link RNAs. So one of the things that I think people have been wondering is, like, why are link RNAs so important, especially in cis? There's many reasons you could imagine this important, but this work from Jesse Ingritz really stood out. And Sophie Condoz actually sent me this paper where it shows that the transcription of this link RNA adjacent gene is going to change this neighbor. What you can see here is when they delete this promoter region for this link RNA, the expression of BEND4 drops substantially. And this is divergent to BEND4. So as we'd predict, the expression of this link RNA is probably helping to support the activity of an expression of BEND4. And the same thing happens for EOMs when they delete this link RNA, suggesting that that potentially it is the divergent promoter that is helping then to increase the expression of both of these genes. In a different type of system, recently, this caught my eye. There was a identification of a de novo deletion in a severe case of a neurodevelopmental disorder. This gene called chaser, or this link RNA called chaser, which is upstream of CHD2, was found to be lost. This results in, again, a severe developmental disorder. When they went and looked at the protein levels, it's known that CHD2 loss can cause severe neurodevelopmental disorders. But actually, in the case of this individual, there's an increase in the CHD2 protein when you lose this upstream link RNA, which is exactly what we predict from our model of upstream dominance, that this link RNA is probably helping to regulate and reduce the activity of CHD2 expression. Which I think is an interesting model and something to be interesting to follow up on. But I would like to also highlight what I've showed you so far is that we have a lot of evidence that supercoiling could be the mechanism here, that this is our model and data are in alignment, but I haven't shown you any data that this is actually supercoiling. We'd like to get that. It's very difficult to measure supercoiling in vivo, and we're working towards that. Additionally, we think that then probably nucleosome structure is changing, and potentially also then the marks across these systems. We haven't looked at that yet, but it's something we're interested in doing. And then finally, we think this will really be important, or we've already seen that it's had a huge impact on our ability to design circuits that can work in primary cells, and that's essential for us to be able to guide these cell fate transitions. So overall, this is part of our model in terms of thinking about cells are integrating all of these chemical, physical forces as they go through cell fate transitions. Therefore, we need to think about how we design them in our models and also in our tools for synthetic biology. This is, I showed you one vignette of the work that we're working on, but they have a lot of people on the team who are working on a range of other things. I'm really grateful for the work of Chris Johnstone, who pioneered the supercoiling work, Casey Love, who's been did all the all-in-one system, and for our collaborators, especially Ross Jones, who's been great, our funding sources. And one thing that's been great at being the Boston area, we have a really good cohort of mammalian synthetic biology folks. And this year we actually have a meeting coming up. And if you're in this area, and even if you're not, I would encourage you to come out to our international mammalian synthetic biology workshop. We have a great lineup of speakers. And one thing I think we know is important in the field is to have more folks who are interested in gene regulation and epigenetics joining and thinking through how do we build gene circuits that make sense in the context of what we know about gene regulation. So the abstract deadline is April 15th, and this will be August 6th and 7th in Boston. So with that, I just want to say thank you so much, and I'm happy to take any questions.
Thank you so much. That was fantastic. It looks like we already have some questions coming in from the audience. So for the sake of letting people ask their questions, I will go ahead and call on Berkeley Grider has his hand up. Do you mind unmuting yourself if he's around? Oh, okay. Or did he?
Hey, okay, now can you hear me?
Oh,
Perfect.
Okay, thank you. Yeah, so I'm just curious, you didn't talk at all about the nature of RNA pol 2 as a molecule that sort of goes in both directions. And I'm
yes.
wondering if the sort of antisense RNA pol 2, which is there, does it, in the case of divergent genes, do you think that the RNA pol 2 that's moving in the other direction ends up making it to the nearby gene that's pointing in the other direction, or contributing at all directly?
So this is a great question. We'd like to be able to answer that question. It's other than doing sort of single molecule work, I don't know that we can answer that exact question. The thing that I think we'd like to be able to do is potentially take and connect promoters that require different polymerases and ask the question, can we get loading of different polymerases, in the sense that one gene is RNA pol 2 and something's like pol 3.
Right.
So you can ask generally, how much of these effects are due to RNA polymerase 2 loading, going in one direction, sending another one the other direction, or working in both, probably not working in both, but recruiting more of the same. And generally thinking about how much of this is just due to the binding affinity changing, again, versus recruitment. Those are hard to answer, but we're trying to set up some systems where we can answer those questions.
Very cool work. Love it.
Thanks, Berkeley.
Yeah, I have a lot of questions too, especially like about topoisomerase, but I want to let like Ben, he has a question, I know, so I'll let him go ahead and do that.
Hi, Katie. That was fantastic. A really interesting talk. Lots of questions. My question was along the same line as Berkeley. I was struck by, as you're talking about this, the divergent setup, just because like when we do kind of nascent RNA profiling with something like ProSeq, almost every promoter in like a human or a mouse cell has some level of divergent transcription. And so, like, based on your model, do you think this is something that like has evolved for promoter function to help with, like, this would in theory help the supercoiling?
Right. So, I mean, I think probably part of that is you're going to have some amount of, you're always going to have some recruitment, and potentially it could the landing could happen on either strand, right? So that we don't have a bias in terms of where it would land and then in which direction. So you'd expect that this is something that generally is a mechanism where you just have more loading. So our mechanism, I guess, saying specifically, any transcription potentially is going to have, you're going to get at those promoters, you're seeing some amount of RNA pol 2 probably going in either direction because it can load on either side. So that at least that makes sense, would make sense to me. I know that there's some promoters out there that do seem to be more directional. And I think one of the questions that we have and really should answer is, when we build some of these really like Frankenstein type synthetic promoters, which type are they and what are they doing? Those are questions we're working on answering even now. Mapping the isoforms that come off of these synthetic circuits is something that I haven't seen anybody do, and we want to do, like, very much. And then also asking, where is RNA polymerase loading across here? Where is it getting stalled? We need to build these in specific sites where we're not going to get swamped out by endogenous regulation and transcription. Most of the safe harbor sites that people will put synthetic circuits into are in the middle of introns, which makes it really challenging then to dissect out what's going on with your synthetic gene versus everything else that's happening in that locus. So we're thinking about where to put things so that we're not parked on a highway and getting run over by all of those native processes. But these are questions we want to answer, and we know we're sort of scratching the surface in terms of, like, we have these outputs that we can see at the protein level, but there's so much more we can know at the RNA and then the nuclear structure, and then also RNA polymerase like loading.
Yeah, and I'll note that a lot of what we see is probably not the same pick being loaded in one spot and going in either direction.
Yep.
But they're far enough apart. They're two picks going in. Yeah.
So it'd be two different. And I guess the question there is, how far can they track before they fall off?
Yeah.
Is it hundreds of bases or is it thousands of bases?
Mm-hmm. Yeah,
and
it's great. Like, a lot of them do terminate quickly. Like Hanukkah from our lab had a really cool paper looking at that exact question of what determines the non-coding PAL2, how far it goes and how far it can elongate?
I should probably look at that. I know we've spaced them fairly significantly in some cases and seen that I think that we still see the effect, but it's unclear because you're also adding in a lot of quote neutral sequences.
Mm.
And it's like, what is that? For what we see in the zebrafish genome, and then also for the examples of these link RNA, the effects, I mean, you know, you're talking about tens of kb, it's pretty large.
Oh,
So
wow.
I think that's those systems we think would be good to look at and maybe parse from just looking at supercoiling, because we think the distance would be, you wouldn't have that effective loading. But then you're tied into a native system where you don't necessarily know all the regulation that's also wrapped in there, which is why we try to build up, thinking we're somewhat orthogonal.
Awesome. Thanks.
Yeah.
Very interesting things to think about. I know we're like a little over time, but there is one question or a couple questions in the Q&A. I'll go ahead and ask one of them from Edward Groh. He's asking about what do you think, how do you think this potential supercoiling inhibition mechanism could be affecting the like head to tail gene arrays like DUX4?
I don't know about DUX4. The one that most comes to mind is actually the Hawks locus. Where Hawks is all collinear. And interestingly, so if you think about the collinearity of this, right, the genes that start on in Hawks are actually the ones that are furthest downstream, and then you have subsequent activation coming basically up. And so to my mind, that makes sense that you would have then the added support of supercoiling silencing the downstream gene. The Hawks genes show this what's called posterior dominance. So as you get to these more upstream genes, it's really difficult then to reactivate any of the lower number Hawks genes. So in my mind, that's actually a very great and stable way of building some of these arrays. Whether that's true or just a really good story, I don't know.
Yeah, but we can test it soon. I like that. That's really cool. Thank you so much. And we're past one now, so I think we will go ahead and wrap up. But Ben did post the link to the workshop in the chat, so in the last 30 seconds, go ahead and grab that before we close, or check Katie's Twitter. She's retweeted it recently. So everyone, thank you very much for joining us this week, and thank you so much, Katie and Xing, for very, very cool seminars today, and we'll see you all in a couple weeks.
Thank you so much.
Thank you.
Thank you both for the seminar. Bye.